Today we are talking about DNA replication, transcription, and translation. Our parent strand of DNA will be replicated and create new daughter strands. These new strands will either be sense strands or antisense strands. We will look more in depth on how this happens and the process our DNA constantly goes through. Semi-conservative replication. The replication starts with two antiparallel DNA strands unzipping. They grow in the carbon 5 to carbon 3 and read in the carbon 3 to carbon 5 direction. They unzip the parent strand due to the enzyme helicases on the hydrogen bonds between the nucleotides. These enzyme helicases separate the hydrogen bonds. Single-strand binding proteins make it easier for other enzymes to function properly on the DNA strands. Sometimes there's a DNA gyrase that will relax the coils when unwinding the strands. This creates two separate strands with unpaired nucleotides. Free-floating nucleotides then pair with their complementary bases. This is called complementary base pairing, in which adenine with thymine and guanine with cytosine are paired.
Replication fork. This is where the replication of DNA occurs. In the replication fork, there is a leading strand where it is replicated continuously, and a lagging strand which is replicated discontinuously. These work in opposite directions. The lagging strands form in sections called Okazaki fragments. DNA polymerase III begins the formation of Okazaki fragments. Primer is the starting point for synthesis, and RNA primase synthesizes it in elongation, which is a primer sequence, and RNA nucleotides, which is later replaced by DNA nucleotides that are added together. Nucleotides that are added to this strand are deoxynucleoside triphosphates. DNA polymerase I replaces primer with DNA. DNA ligase helps connect the sugar phosphate backbones from the lagging strand to form a single DNA strand.
The RNA polymerase, antisense DNA, and growing transcript are inside a transcription bubble, which develops when the DNA starts to separate. RNA will make a copy of the exact sense strand, but replaces the thymine with uracil. mRNA is the transcript that carries the copy of the DNA. Phosphorylation binds phosphate groups to different molecules. It uses kinase, which can transfer a phosphate group from ATP into a protein.
Ribosomes are where the proteins are made. Each consists of a large and small subunit that joins amino acids. The subunits are composed of ribosomal RNA molecules and distinct proteins. In initiation, codons, which are sections of three bases of messenger RNA attached to specific sections of transfer RNA. Transfer RNA transfers one amino acid to a ribosome to create polypeptides. A loop on the transfer RNA is an anticodon, which pairs with the codons from the messenger RNA. In elongation, transfer RNA moves amino acids to messenger RNA. Transfer RNAs are bonded to codons at the A site, the entry site in a ribosome. Then it moves to the P site where the polypeptide bonds are catalyzed. Later, it leaves through the E site during translocation. In termination, three stop codons appear at the A site. The release factor links the transfer RNA to the polypeptide chain, releasing it from the ribosome. The translation is completed.